there, happy little molecules. Today I'm going to be showing you how to figure out the structure of the molecule C6H12O2 given the molecular formula, IR spectra, carbon-3 NMR spectra, and the hydrogen-1 NMR spectra. Before we dive in and start analyzing our graphs, let's talk a little about requisite skills for interpreting what we see. The first thing I want to keep in mind is the degree of unsaturation. Calculating this will allow us to insert the correct number of double or triple bonds in our molecular jigsaw puzzle. What is the degrees of unsaturation? The short answer is that it is the difference between the number of hydrogens there would be in a saturated alkane where all bonds are single and the predicted number of hydrogens that we are actually seeing. A du of any non-zero number is the number of pairs of hydrogens that are missing. This number is equal to the number of bonds that are needed to make up for the missing hydrogens. The du formula is taking 2 times the number of carbons in your formula plus 2 minus the number of hydrogens in your formula, and then everything divided by 2. A du of 1 would mean that 2 hydrogens are missing, and in order to make up for these absent hydrogens, we will need to create 1 double bond. If we do not do this, we end up with lone pairs. Let's define anisotropy and talk about how it affects polarity. Anisotropy is the resistance to being pulled downshift by the electromagnetic attraction to another atom due to having lone pairs, which is also known as shielding. Anisotropy depends on polarity. If 2 atoms are attracted to each other and have the same electronegativity, there is no shift in their data points. However, if there is a difference in electronegativity between the 2 atoms, the more electronegative atom will pull the weaker atom downshift. This pull may be weaker if the more electronegative atom has lone pairs, which act as a repulsive shield to the weaker atom's valence shell and prevents the weaker atom from getting too close. Anisotropy and polarity can affect our data points in 2 main ways. Atoms bonded to the carbonyl carbon appear significantly downshift from the range at which they are typically predicted. Atoms bonded to oxygen, however, only experience a slight downshift due to having lone pairs. One last note. If we have symmetrical structures or functional groups in an atom, each unique structure will only report as 1 data point on the graph. This means that there may be multiple iterations of that part of the whole molecule that are not repeatedly represented on the graph. Cyclohexene is a perfect example. All carbon atoms have 2 single bonds to other carbon atoms and 2 hydrogen bonds in the same layout. There is only 1 unique structure in this molecule. Let's take a look at our data. In the next few slides, our top graph will be the data presented from the actual molecule. The bottom graph is a key containing the peak characteristics of different molecular bonds and a range in parts per million where they are detected. There are 2 distinct peaks on the left above 1500 parts per million that we will focus on for the IR spectra. Everything below 1500 is referred to as the fingerprint zone. This is beyond the scope of our class and can be tricky to read correctly. Let's start with the peak on the right. The data point is at about 1750 parts per million. The only value for that reading in the key is a double bonded carbonyl carbon. This seems to be right on track with the key's prediction. We know from the du that this reading cannot be the triple bonded carbons, and nitrogen is not in our formula. The carbon double bonded to another carbon could be an option if the parabolic shape of the peak was much more sharp and narrow, and not instead reflecting the oxygen's characteristically wide spacing. The leftmost peak is very narrow, and at the tip of it is located at about 3000 parts per million. When we compare the data point to the key below, we notice that the suggested structure is trigonal planar and contains double bonds. Since we have calculated du to be 1, we know that there can only be 1 double bond in this entire molecule, and that the double bond belongs to the peak that was measured at 1750 parts per million. Here we have our carbon-13 NMR spectra. This is the only carbon isotope that can absorb radio waves in a magnetic field, making it perfectly detectable under NMR spectrometers. Note that in the range for this spectra is only 200 parts per million, unlike the 4000 for IR. Let's start from the left. The first peak is at 175 parts per million. On the key, 175 parts per million clearly reports to be a carbonyl carbon double bonded to an oxygen atom and also single bonded to a different oxygen atom. The next peak is 55 parts per million. This peak, according to the key, can be either an sp3 tetrahedral carbon single bonded to an oxygen atom or a carbon single bonded to a nitrogen atom. Since there is no nitrogen in our molecular formula, the closest suggestion is the former. It appears that there is a possible slight downshift from the predicted value. We should keep this in mind when trying to figure out how close the carbon is to a carbonyl group or an oxygen atom. The next peak measures at 45 parts per million. The same explanation applies as did for our value at 55. The next peak is at 25 parts per million. This falls within the 0 to 40 parts per million range and is an sp3 tetrahedral carbon. We must note that this structure does not list what the carbon is connected to. Thus, it could be connected to either other carbons or other hydrogens, but not a heteroatom like oxygen. The same explanation applies with our last data point, 21 parts per million. Now, let's analyze the data in our hydrogen-1 NMR spectra. Let's look at the data point at 3.6 parts per million. The key suggests two possible structures, one of which contains nitrogen, which is not in our molecular formula. The other value is the nearest on the key, which is an sp3 tetrahedral carbon that is single bonded to both one hydrogen and one oxygen. Since this carbon is sp3 hybridized, it means that it has 4 single bonds. The integral number given to us above the peak, 3H, tells us that there are 3 hydrogen atoms attached to the target molecule. You will also notice that there is a single peak for this data point. The number of peaks for any data point is used to calculate the number of hydrogens on atoms neighboring the target atom. To do this, we use the n+1 rule, which simply states that there will be one more peak than number of hydrogens on the neighboring atom. This target atom must then have 0 hydrogens on its neighbors. The next data point is at about 2.45 parts per million. The integration number is 2H, which means that there are 2 hydrogens on the target atom. It too has an sp3 hybridized tetrahedral structure like our previous example. There are 2 peaks, called a doublet, and by the n+1 rule, has only 1 hydrogen on its neighboring atom. The next peak is at about 2.25 parts per million. The key suggests that this value lies in the range of that for a carbon bonded to a nitrogen, which is not in our formula. This means that the data point value is shifted way up at about 1.5 due to some heavy shielding preventing it from being at its normal range. We can justify this data point is not another sp3 hybridized tetrahedral carbon like our last couple of readings because there are 7 peaks that suggest it has 2 neighbors, each connected to 3 hydrogens, which also suggests that some massive shielding is going on here for our target carbon. Lastly, we have a doublet at 0.9 parts per million. This one is a little unusual because none of the atoms in our molecular formula can hold 6 hydrogens. Instead, this is what is known as a doublet of doublets. There are actually 2 target carbons being read, and each has 3 hydrogens attached to it. Also, each of these carbons must only have 1 neighbor, and that 1 neighbor has only 1 hydrogen attached to it. We know this neighbor is shared and not 2 separate monohydrogenated neighbors because our last slide perfectly complements this slide's molecular shape. Here are all the structures we have figured out so far. The X's indicate unknown connecting neighboring atoms. Here is a table of all the structures we know, along with their accompanying information. This is a handy helper for solving a molecular structure problem using the multiple spectra as clues, as you can compare the magnitude of shifting in relation to how the structures might piece together. We piece together the obvious molecules first. Written in green are the atoms determined by the hydrogen-1 NMR, and in gray are the reported neighboring configurations. This is the easiest piece that we have because the exact match in shape and unusual number of hydrogens attached to the neighboring atoms. Here we attach the final piece for the spectra. Now we attach the carbon-13 NMR functional groups. Since the X's can attach anywhere, we look back at our table to make sure that we are attaching it to the correct molecule and that the correct number of neighboring hydrogens are maintained. In this example, the X's refer to existing neighboring molecules. The C is also not a new carbon, it is just a placeholder. What we really focus on here is a single bonded oxygen that tags onto the carbon. Here, we can see that both the carbon-13 results and the hydrogen-1 results both reported on this structure. The carbon-13 was able to give us a tiny bit more data that there is a single bonded oxygen on the carbonyl carbon, making this an ester. Using the X as a placeholder again, we tack on the carbonyl group. Finally, we add the last group that we have detected from our IR spectra onto our molecule. There is only one viable space left, and it is on the carbonyl carbon. This would explain why its data point was shifted about 200 parts per million downfield. The resulting structure is 2-methylpropyl acetate. We solved the mystery. Congratulations! You're not confused anymore.